All right, guys, in this unit, we're going to be talking about the cell membrane. And you might be wondering why we are spending so much time on the cell membrane, just one cell part. Well, there is a lot that happens at the cell membrane. And so we have to study the cell membrane so that we can help us in different situations. Here's an example where we are taking white blood cells. We are causing them to put proteins in their membrane. And then we can take these cells, put them in a person, and those cells can fight cancer cells. In diabetes, what is wrong oftentimes is some of the proteins in the cell membrane aren't working. And so then we have to figure out a way to fix them. We have to learn a lot about membranes so that we can design molecules to break holes in cell membranes so that we can kill particular cells that are giving are causing an infection. We have bacteria cells that constantly are evolving and our drugs no longer work. So we have to keep finding new ways that we can destroy them. And one thing that we can target is their cell membranes. So there's a lot of things that happen with cell membranes, and that's why we spend so much time with them. And we've already talked a little bit about cell membranes. And we've mentioned before that cell membranes help regulate what moves in and out of the cell. Now, some molecules can cross through that cell membrane. Some molecules can't. So that's why we say that the cell membrane is semi-permeable. So some things can go through it, but some things can't, okay? And we'll see that cells differ by what they can let in the membrane or out of the membrane. And they can differ because they can make different transport proteins. So this cell, if it makes a specific transport protein, then it can allow sodium to move in. Whereas other cells don't make those transport proteins. So sodium doesn't come in, and those cells are going to have a completely different task for the organism. Now, we're going to talk about how the membrane allows cells to communicate. And before we talk about this diagram, let's show you what I mean by cells can communicate using their membranes. So here we're looking at a bunch of cancer cells in an organism, and they're dividing out of control. And cancer cells will release molecules that are foreign to the body. And those molecules can activate certain types of white blood cells. Now, this white blood cell, when it becomes activated, is going to activate this other white blood cell. And you can see here we have two cell membranes with proteins embedded in them, and they're going to touch. And when that happens, it'll activate molecules that are in the cytoplasm of this cell. So the cell is receiving information, and then the cell is going to respond. In this case, the response is going to be this activated cell is going to divide to make more. And these cells are cells that can bind to cancer cells and can cause them to die. So we saw that cells communicate. In this case, cells are communicating simply by touching. We also have situations where cells can communicate because they can release hormones that can travel and go to a different cell. So for example, let's say that this is a hormone, and this hormone was made by the pituitary gland. The pituitary gland is at the base of your brain. So these pituitary cells made and release these hormones, like this could be a growth hormone. And this will travel to a cell that can respond to this growth hormone. So let's say that this is a bone cell. And once this molecule binds to a protein in the membrane of this bone cell, then the bone cell is going to divide. And you're going to get more bone cells, and your bone is going to elongate, and therefore you're growing. So you're growing simply because we had cells in different areas of the body being able to communicate through chemical signals, and oftentimes those are hormones. And then kind of along the same lines with that is cell membranes help you maintain homeostasis. Now, homeo or homo is a prefix that means same. If you're trying to maintain homeostasis, then what that means is that you are trying to maintain a constant, non-changing environment. There are just some things in our body that we have to carefully regulate, and we have to keep very specific levels. And if our levels get off, then we're not going to feel very well. our body's going to have to respond to bring those levels back to where they should be. Let me hit this focus button again here, see if I can get this to focus a little bit better. All right. So some things that have to stay constant in the body. Let me turn on the lights. That we have to maintain homeostasis is water levels. And cells can detect with their membrane if your water levels are going down, or if you're getting too many solutes. You have to maintain blood pressure. And we have cells that can monitor blood pressure and cause a response if your blood pressure is too high or too low. We have to keep the right amount of glucose in our blood. Too high, too low, and you can pass out, because then your cells and your body systems aren't able to function if they have too much glucose or they're lacking too much glucose. Our pH, our oxygen levels, our CO2 levels, and then temperature. We all know that we have to keep a very constant body temperature. So our cells are able to detect and respond. And lots of times that detection, that response has to do with something that happens at the membrane. So for example, in this case, we have a protein hormone, and this insulin, and insulin is made by beta cells. So I'm going to put a beta cell that is a long ways away from this target cell. The beta cell is in the pancreas, and it can produce insulin. It releases the insulin, and it does it by vesicles. So I guess we should put it in a vesicle, and send it to that membrane, because insulin's a really big protein. And it then releases or secretes that protein. It moves into the bloodstream, and then eventually that bloodstream carries it here to this target cell. Whenever that insulin binds to this target cell membrane and this protein that's in the membrane, and that causes the cell to insert other proteins in the membrane, and then the glucose that's coming out of your blood can go into the cell, and it can be chained together and stored. And you might remember that when we chain glucose together to store it in our cells, it makes a molecule that is glycogen. To tie in some of the stuff that we learned in the last unit, okay? So if we have too much glucose, cells release insulin, and then insulin gets brought into the cells, and now our glucose levels become more at a normal level in our bloodstream. So we have to maintain homeostasis, and we use cells and their cell membranes and the different processes that can happen there to do that. Next, we're going to take a look at a more in-depth look at what our cell membranes look like, what they're built out of. And again, we've already talked a little bit about this. So there's really three main types of molecules that are in your cell membranes. Your cell membranes are primarily made of phospholipids. And we also find that there's going to be some proteins. We've already shown you some proteins in some of these images and in that last video. And we also have cholesterol. Cholesterol sometimes is thought of as a molecule that's bad, but we have to have cholesterol in our cell membranes. Now, these molecules come together because they have polar and nonpolar regions. We've mentioned in the biochemistry unit that phospholipids are the lipids that primarily make up our cell membrane, and those heads are polar, and polar will interact with polar. Remember, water is polar, so these heads will be attracted to polar water molecules. And since they are, we say that they are hydrophilic. Whereas these tails, they were fats. And most fats do not interact with water because they are non-polar. And molecules that will not interact with water, we say are hydrophobic. Okay? So because phospholipids have a polar and a nonpolar part, they come together to form this membrane as a bilayer. Now, on the inside of our cells, we have lots of water molecules. Our cytoplasm is mostly water. The outside of our cells is mostly water. And if you put a bunch of phospholipids in this aqueous, water-filled environment, then they'll come together and they'll form this bilayer on their own, just simply because these polar heads will interact with each other, and they'll also be attracted to water molecules. So they'll arrange themselves more on this outside of this interior where they are interacting with these water molecules. These nonpolar parts, will be repelled by water. So if there's a water molecule here and then there is a phospholipid where, it comes in contact with these fatty acid chains, it'll flip around. So that polar part will be facing the water and the tails will be away from the water. So we end up with these areas that are polar. Now I'm going to go ahead and shade all of these molecules here, and the parts of them that are more on the outside of the membrane, I'm going to lightly shade them, because these phospholipids and then these proteins here, these parts of those molecules are polar regions or polar areas. So same over here. These parts that are facing outward where all these water molecules are, just shading it in nice and light. So even this part of cholesterol, this upper part right here, that is a polar region. Whereas our interior of our membrane, all of these parts that we see here and here and here, those are all non-polar areas of those molecules. So if we send a protein to the membrane and the protein is mostly a polar protein, it's not going to embed here in those phospholipids. It's going to adhere and stay more on this outer part of the membrane. So phospholipids and all of our proteins arrange into this phospholipid bilayer simply because they all have polar and nonpolar regions that affect where they're going to go and how they're going to come together at this membrane. Now, we're going to learn more about the proteins and their different jobs, but let's just take a look at cholesterol real quick. And what is the role of cholesterol in the membrane? So cholesterol is also a fat. It's a steroid, and it has polar part and it has a nonpolar part. And if you have ever taken, like if you've cooked food, let's say pot roast, and it has a lot of fat in it, and you put it in a refrigerator, whenever fats get cold, they become very solid. When fats get really warm, they can become liquids. And we have to be careful here because we do not want this fat layer. Remember, phospholipids are lipids. We don't want this layer to become too solid that nothing can pass through it. We also don't want this phospholipid bilayer to become too liquidy and then it just breaks apart and it can't hold the contents of the cell. So that's where cholesterol comes in. It really stabilizes the structure of the membrane. It doesn't allow the membrane to become too liquidy, and it doesn't allow the membrane to become too much of a solid. So it keeps it very fluid. Now, since we have all these different parts that come together here to form what is the cell membrane, we have a name for this model of the structure of the cell membrane. It's called the fluid mosaic model. Okay, so fluid because all these little parts are individual molecules and they can move around kind of independently of one another. So I'm going to kind of fast forward here so you can see that it's a fluid structure. I mean, they can move around. Molecules can kind of push them apart, yet they still stay together as a kind of a solid there. So it's a fluid structure, but also it's made of a lot of different pieces. And that is what a mosaic is. If you're familiar with a mosaic, like it's a piece of artwork where you have all these individual little pieces that have their own unique properties. They all come together and they form this bigger piece of art. So here we have all these different pieces that come together to form ultimately what is this cell membrane. So it's called the fluid mosaic model. All right, now I want to touch real quick on some of these proteins and their specific jobs. Before I do that, let's watch this video and let's look at what some of these different proteins look like. We are moving along the surface of a cell membrane, so you can see that we have these circles. Those are the heads of the phospholipids. And we see, I'm going to pause here, some of these proteins, it looks like they have this, extra structure, almost like a tree branch sticking off of them. So that's one type of protein. And we have some other proteins, though, that look a little bit different. Let me fast forward a little bit here. So this protein inside the membrane, it doesn't have any of these branches off of it, and it's a different type of protein. And we can see that this protein has a shape to it that molecules can bind to. And this would be a hormone, and it's binding to this protein. And whenever it binds, it changes the shape of this protein. And this is what signals a cell to respond. We talk about how cell membranes allow cells to communicate. Well, now this cell just got a signal, and it's going to be able to respond in some way. We just saw a couple different types of proteins in that video. So back to our notes here. That protein that we just saw that a hormone or a signaling molecule bound to is called a receptor protein. Receptor proteins can receive signals. They can have other cells hook onto them, or hormones can travel and bind to those receptor proteins, and it causes the cell to respond. And usually, again, a response is necessary because you need to change something in the body. You need to work to maintain some homeostasis. The other molecule, or the other protein that we saw there, I'm going to draw it here. And it looked like there's these branches. So I'm going to draw kind of this branch structure coming off of this protein. Now, this is called a marker protein. If you think of, we use a flag, like, decades ago in times of war. If we claimed an area, we would put up our flag, and we would say that would indicate, like, hey, this is, the people that are in this area, they are friend, they are not foe. And here's our flag that shows that we are not the enemy now in this location. These marker proteins are a way for your body to know what cells are yours and what cells are not. Because most of the time, whenever you have cells that aren't yours in the body, like bacteria and fungal cells, then those cells are foreign cells, and they can lead to infection. So we have marker proteins that identify which cells are ours, and other cells will have different marker proteins, and our cells can use those proteins to identify that those cells are foreign. They need to be destroyed. Okay? Now, a little bit about these marker proteins. There's two parts to them. This part down here is actually the protein. But in the last unit, we talked about how, like, the rough ER modifies proteins. This part right here got put on by the rough ER. So the ribosome made the protein, the rough ER got it, and then attached this part to it. And this particular marker protein with this attachment to it would be called a glycoprotein. Now, if you remember, we've talked about a word before, glycogen. Glycogen was a string of glucose molecules. So each of these little circles right here is a glucose molecule. So in the rough ER, that rough ER can take the protein from the ribosome, and it can attach a strand of glucose molecules to it. And remember, a strand of glucose was called glycogen. So that's why these are called glycoproteins. This protein is part protein, and it's actually part carb to do a job for you. The next protein we're going to look at is a transport protein, and it's nice because it's named after what it does. Transport proteins can transport molecules through the membrane, and on the previous page, we drew one right here. And so we had a protein that has an opening in it, and let's go ahead and label this was a transport protein. Molecules can go through transport proteins if the molecules and the proteins are the right shape and size. And then finally, we have some proteins that are attached to the membranes, are enzymes. And we've talked about enzymes before. Enzymes are proteins or molecules that can assist in chemical reactions. I'm going to turn my lights back on. And we have built posters that showed that here we can have enzymes, and enzymes have a certain shape that the substrates can bind to. So in they go. And this enzyme, so I'm going to put an E for the enzyme, can then, in this situation, can attach them together and assist with this reaction. And now they are a different molecule because they've been bound together by that enzyme. Okay, so some enzymes are free, like in the cytoplasm or outside of cells, but some are actually embedded and attached to a membrane. So let's take a look at some of the ways that we can diagram these proteins. So this is showing you the phospholipid bilayer. They're just not showing you that, there is actually a bunch of phospholipids right here. It's just a little bit easier sometimes to diagram that bilayer like that. And in order to determine what a protein is, you have to, show some features of it or show some action. If we see molecules moving across the membrane through this structure, that must be a transport protein. If we see a protein that has this carb chain, so let's indicate this is a carbohydrate chain. I'm just going to write carbo chain there. Then this must be a marker protein because the shape of this part is utilized by other cells in your body to identify that this cell that you have here with these proteins in the membrane is a good cell. It's not a foreign cell, so do not destroy it. And then we know that this is a receptor protein, one, because we can see that something is now happening. There's some kind of signal going off in the cytoplasm of that cell, and it's all because we had this molecule come to the cell, bind to it, and then activate that signal. So we have this protein is receiving a signal that has been sent by another cell. And then here we know that this must be an enzyme because we can see some change happening between molecules. We have, let's write, molecule X is going into the enzyme, and it is now turning it into molecule Y. Remember the substances that go in, so we could call X as the substrate, and then what comes out, we then call the product. So Y would be the product. So if we see something like that happening, like we diagrammed on our posters, we must know that that must be an enzyme. You can also take a look down here and probably figure out that we have a receptor protein. These are different marker proteins. In goes one molecule and out it comes as a product. So these must be enzymes. And then finally we have molecules moving through. So then we got a transport protein.